It's great to be back at Cornell. Every time I come here, which is not as often as I'd like, I always wish I came more often because there's so many great people here and so many friends. So I will try and come more often, but I think it's been about 10 years since I was last here. Yeah, so I'm going to talk about development, which is what we study, very similar to the work you know from Mike's lab. And you know, I'm really fascinated by this idea that genes control development. It's a very simple idea, but you know, development is obviously very complex, the generation of form of three-dimensional structures. But the idea that we can tease this apart by finding individual genes that control specific aspects of development is something that I find very attractive, because I want to know how things work. Ever since I was a kid, I would be taking toys apart and breaking them, you know, and finding out how machines work. So I want to know how plants work, and specifically how the morphology is generated. And you know, potentially we could use that to improve crops. I don't know. Maybe I'm very naive, but you know, I think you know, we should always think about potential applications of our work. So very much like Mike's lab, we study the meristems, the shoot meristems, and I always sort of assume that everybody knows what a meristem is, but I know most people don't. But anyway, the meristem in maize, which is what I'll talk about today, is found right at the base of the seedling, not up here, which is where most people guess it will be, or some people think it's here, but it's actually right at the base. But if you're very patient and dissect away all the leaf primordia down here, then you're rewarded by this amazing structure, the shoot meristem, which I think is beautiful. And it's this dome of cells, stem cells, that throughout the life of the plant generate all the organs that are required for growth and development. So that's a scanning EM, because the meristem is pretty tiny. It's about a 1/10 of a millimeter in diameter. But this structure obviously is critical for plant development, because it makes all of the structures of the shoot, including the leaves, the axillary meristems, which make the inflorescences, the ears, and also the tassel. So it's a critical structure, and it's something we can analyze and understand using genetics. Actually, 25 years ago, when Mike and I were in Berkeley, I remember Ian Sussex told me that the meristem was a black box, and it was a killer project to put a postdoc on that, because you couldn't understand it. It was so mysterious. But you know, he didn't, although he wrote the book on meristems, Steves and Sussex, he didn't use a lot of genetics in his lab, and I think he came before the era of genetics. So genetics is the key, really, to understanding this. Okay, so what you should also realize is the meristem doesn't also make leaves, but of course, after transition to flowering, it produces the reproductive organs, reproductive structures. And so these are also shoot meristems. And so you have the axillary shoot meristems make the ear primordia, that make the ears, and the apical shoot meristem produces the terminal tassel that is similar in structure to the ears, except it has branches, of course. The tassel is branched in maize. But these are all meristems. These are all meristem structures. And we can study genes that affect, you know, all of these different stages of development, but we tend to focus on the ear, just because that's where the phenotypes that we work on are more dramatic. So we look a lot at ear primordia, and these things that we affectionately call baby ears, right? So that sometimes gets us in trouble with FedEx when we ship packages labeled baby ears. We get some strange reactions from them. But anyway, yeah. So you're all familiar with the ear of corn, of course. You've eaten this, and everybody loves it, I hope. But we look at the ears when they're about this stage. This is 2 millimeters, about the thickness of a quarter. And at this stage, already, most of the morphogenesis, the initiation of all of the structures has occurred. And on the flanks of the apical meristem, you have all of these primordia forming. These are called spikelet pairs. Then they form spikelets and then florets. And this is what produces the seeds. So the meristem at this very early stage is already making this very regular rows of primordia that will make the regular rows of seeds. So the morphology is set up very early. So there it is again. And these are all scanning EMs, by the way, and they look impressive, but these are really easy to take. So this is just live tissue. We put it in this SEM, and 2 minutes later, we have an image like this. So you should play around with that. So we've characterized a lot of the genes that are involved in ear development, and I'll just give you an outline of some of them. I mean, there are a lot of different things going on here. So in the inflorescence, there are leaves that are produced, but they're actually suppressed. Their growth is suppressed to promote growth of the meristems, and that's a class of genes called tassel sheath, which Clint Whipple is now focusing on. He's cloned the first one of those in my lab, but he has his own group now working on those. The axillary meristems are initiated, and that's a lot of the genes involved in that process are involved in auxin signaling, as we know also from Arabidopsis, and work from Paula McSteen and Andrea Galvaudy has worked a lot on these genes. We work and continue to work on genes that impose determinacy. So this is really a branching, series of branching events. Actually, 3 rounds of branching produce the florets, but then that indeterminacy has to be turned off. So determinacy has to be imposed. And that is characterized by a class of genes called the Ramosa genes. And we're working on those too. But for today's seminar, because I know it's lunchtime and everybody's going to get hungry, I'm going to focus just on one group of genes. And those are the ones involved in maintaining the stem cells right at the tip. And these are the very famous genes known from Arabidopsis, from the work of Elliot Meyerowitz and many other groups, the Claddagh genes and the Wuschel genes. So that's what I will focus on today. Now, we've known for a really long time, actually since the 1920s and before, that the meristem has some kind of program for controlling its size. So this is, the meristem is a structure of proliferating cells, but the cells are not proliferating out of control, of course. It's very carefully controlled. And Mary Pilkington did these very nice experiments in the 1920s, where she would remove all the leaf primordia from meristems. I think this is in beans, and then bisect the meristem with a razor blade, and then each half will regrow, regenerate, and produce new meristems that are the same size as the original meristem was. So that the meristem has some kind of internal program to control its size. And we know now from a lot of work, mostly in Arabidopsis, actually almost exclusively in Arabidopsis, that this mechanism for controlling size is through this classical Claddagh-Wuschel feedback pathway. And work on that has told us that there are 2 really important groups of cells in the meristem. Right at the tip, you have the true initials, the stem cells, that we know from clonal analysis. These are the true progenitor cells of the shoot. And then below that, there's a group of cells in purple here, which is called the organizing center. And basically, to understand how the meristem is maintained, you have to understand that there's a crosstalk or a feedback between these 2 groups of cells. So the organizing center sends a positive signal to the stem cells to tell them to divide. Okay, so that you need to promote stem cell differentiation to maintain the shoot. But to maintain this as an ordered structure, this has to be kept in balance. And it's kept in balance by a negative signal sent from the stem cells back to the organizing center. Okay, so mechanistically, that's how it works. And what are the genes involved? Well, in the stem cells, you have the Claddagh genes, as I mentioned. And up to now, most of the work has been done on a couple of leucine root repeat receptors called Claddagh1 and Claddagh2. And their ligand, which is a small secreted peptide encoded by a gene called Claddagh3. So this gene makes a pre-peptide of about 100 amino acids that's cleaved into a 12 amino acid signal that's secreted, and it binds to Claddagh1 and activates signaling from this kinase. And then the organizing center is characterized by this famous homeodomain transcription factor Wuschel, which is expressed down here in those cells. So Wuschel promotes stem cell fate, Claddagh genes repress stem cell fate. Okay, and we know this is important because in nature, in the wild and in our gardens, we see plants where we've lost this negative feedback. And I call this stem cells out of control. The botanical term for that is fasciation. And in that case, the stem cells overproliferate, and you get these amazing convoluted shoots or inflorescences or flowers or fruits. Okay, so tomato, for example, work from Zach Lippmann and Esther van den App has shown that a Claddagh mutant is what gives rise to this beefsteak tomato. Okay, so I had to show this being a Kernel, but also this is a very famous picture from Ithaca, 1929. Actually, I just realized this is the same year that that meristem bisection experiment was done. So 1929 was a good year for for plant biology. But anyway, this is Barbara McClintock, who discovered transposons and later, of course, went to Cold Spring Harbor. Why do we study this in maize? Because, as I said, most of the foundational work has been done in Arabidopsis. Arabidopsis is a fast cycling plant with a small genome. Maize is a slow cycling plant with a huge genome. But still, I think we can find new things in maize. And one of the great things about maize is we have a huge mutant collection, right, that is generated in part by the scientists who developed these transposon lines. And so in maize, we have a lot of Gbeta type mutants, which is the foundation of the work that I'm going to tell you about. So we don't call these Gbeta, we call them fasciated ear. So fasciated, again, is the botanical term for these massively expanded stems. And these mutants, and this is just 5 I'm showing, we have another 20 that we haven't characterized yet, but I thought I would keep it focused. Some of these mutants, as you would expect, we clone them, and they're orthologs of the Gbeta genes, right? This is not surprising. So TD1 is Gbeta1, this LR receptor. Fe2 is Gbeta2. And then others that we've cloned have been different. So we have found new things in maize that were not discovered in Arabidopsis. So Fe3 is a new type of LRR receptor that functions somehow in the Gbeta feedback, but was not characterized in Arabidopsis, but it does function in Arabidopsis. Fe4 is a transcription factor. And I'll talk a little bit about CT2. That's a Gprotein that we think is involved in signaling downstream of these receptors. Okay, so we have a large collection of mutants, and it's really, really great material to work with to discover the genes that control this meristem feedback. Now, another thing I'll talk about today, and this is more recent work, is the idea that these genes may be useful for controlling yield traits. So in maize, there's breeders measure yield traits such as, obviously total mass of corn that's harvested, but they also use more quantitative traits such as this one, kernel row number, which is the number of kernel rows around the circumference of the ear. So if you break the ear in half, you can just count these. So this one has 12 rows. This one, if you count quickly, it has 22. And we have this idea that by controlling the size of the meristem, you could control the number of rows, because you see this is now looking down from the top of the ear primordium, and this is the apical meristem, and then all of these primordia are the ones that are making the rows of seeds. So naively you could say, well, if we can make this meristem bigger, then we could make more rows of seeds, right? So that's what we want to do. So the other thing that's really interesting about kernel row number, of course, is that maize itself is a man-made crop. It's genetically modified by humans over the past 10,000 years, and it's derived, as you know, work from John Dobley and Ed Buckler and others has shown that maize is derived from teosinte, which had only a single row of seeds, and human selection over the course of thousands of years gave rise to the amazing ear of corn that we have today. We know this is genetic, right? So kernel row number is acts as a QTL, so it's heritable, and you can map loci that control it. And we also know this was a gradual process. So primitive cobs of maize found in high altitude caves in Mexico, look like this or this. They may have 8 rows or 12 rows. And of course, more modern varieties that we eat or cows eat have 18, 20 rows, right? So it was a trait that was gradually selected over the course of the last 10,000 years. Okay. So our mutants may have something to do with this. That's what we want to find out. But, our fasciated mutants have a lot more rows, but you see the rows are really disorganized. And the yield of these mutants is very low. Like a farmer would never want to grow this. But, they make too many primordia. It's almost like a cancer. The meristems are proliferating. And in the end, it's making hundreds of primordia up here, but none of them are productive. They just are bored because there's too many of them and they're fighting for resources, right? So we want to try and control that. So the model, our sort of thinking is like this. So if we knock out one of these fasciated genes completely, so zero activity, we have a massive overproliferation, high kernel rows, but very low yield. But if we could make an allele of a gene like this that had reduced activity, like a hypermorphic allele, maybe we could make the meristem a little bit bigger, but it's still organized, and make more rows of seeds around this bigger meristem, and somehow optimize yield or kernel, at least kernel row number. So that's what we'll get to it towards the end. Okay, so we call this like the Goldilocks principle, or the sweet spot, right? Too hot or too cold is not good, but you want to be right in the middle and have the right temperature or right level of gene expression. Okay, so I want to go through one example of a mutant we've cloned, just to give you, oops. Is that me? Yeah, just give you an idea of the process we go through in finding the genes and characterizing these mutations. So this is one mutant, and this mutant is called compact plant 2. So this gets confusing. This should be called fasciated ear or something, but this mutant was discovered in the 1960s before I was born, so I didn't get to name this. And I respect old maize geneticists, so I don't want to change the name, so I keep this name compact plant 2. And as its name suggests, the mutants are very compact. They're dwarfs, but I don't care about that so much. What I care about is they have fasciated ears. So this is the phenotype that I'm really interested in. Okay, so we cloned this gene, and it was probably 3 years of work, but like most people, I just show it in a single slide. And we found that it encodes a protein called Galpha, which is the alpha subunit of the heterotrimeric G protein. So these genes are super famous in mammalian systems because G-alpha is a signaling protein, it's a GTPase, that interacts with a class of receptors that are called, not surprisingly, GPCRs, which stands for G-protein coupled receptors. And GPCRs in yeast and in humans are these 7-pass transmembrane proteins. And these are targets for about half of all pharmaceutical drugs that are developed for humans. So they're very important in the pharmaceutical industry. So G-alpha is the signaling protein that when these receptors are activated, this protein is a GTPase. It will be activated by transferring GDP to GTP, becomes activated, dissociates from its subunits, beta and gamma, and those dissociated subunits go away and start signaling cascades, right? So because these proteins were so famous in mammalian systems, people started working on them in Arabidopsis and made mutants in them and found that they did all sorts of different things. So basically anybody who screened them for a phenotype would find their favorite phenotypes. If you were looking on cell proliferation or different hormones or sugar signaling or defense responses, people found a lot of phenotypes, but there was nothing specifically up to now to do with meristems or development. So we thought we had a unique angle here because we'd shown that this mutant in G-alpha had a very clear meristem size phenotype. Okay, so then what we do all along with these mutants is we try to put the genes into pathways. And we do that by epistasis analysis. And so we make double mutants, and here we made a double mutant between CT2R G-alpha mutant and Fe2, which, if you remember, is one of the Claddagh receptors, one of these feedback stem cell receptors. And we found that the double mutants, and here we're measuring, we did this by measuring meristem size, but we can also do it in a more easy way by counting spikel density in the tassels. We found that double mutants look like the single mutants in terms of spikel density. And so this is what we would call epistasis and suggest they're in a pathway. Now we did something that serves as a control experiment, a double mutant with TD1, which is a different Maize Claddagh receptor, Maize Claddagh 1. Here we see an additive phenotype, and this suggests that CT2 and TD1 are in different pathways, right? They're acting in parallel. Okay, so we had epistasis with Fe2, suggesting these proteins may be acting together. So we wanted to look further at that, and so we made a GFP or YFP fusion of CT2 and fused it in frame with the native promoter with the CT2 coding region. We could image it, and it's on the membrane, which is what is predicted from all the work in mammals. And fortunately, as we hoped and expected, we saw high expression in the meristem. So it is expressed in the meristem. and has some interesting patterns. I don't really have time to go through that. But, so these, reporters, as Mike said, can be very useful for just looking at where things are expressed, but they're also really useful for more functional assays. And so we had a hypothesis that CT2 might interact with this Clavata receptor, and using the GFP as a tag, we could use that for IP experiments, immunoprecipitation. And so we pulled down CT2 with a GFP antibody in an immunoprecipitation experiment. And we actually we already had an antibody to detect Phe2, that's shown here, the specific antibody that detects this Phe2 receptor in the membrane fraction. And we showed that if we IP CT2, we pull down Phe2 receptor. So pulling down the G protein also pulls down the receptor. And we did controls with a different membrane protein, because these membrane IPs are actually very tricky and you can get a lot of background, but we did a different membrane protein, PIN1. When we IP that one, we don't pull down the receptor. So we think it's specific. So our model now is that this G protein is signaling from the Clavata receptor, from Phe2, right? So if that's true, we should be able to see some other kind of Clavata phenotypes in our mutant. Well, we saw it has big meristems, but you can get big meristems for a lot of different reasons. So to see if it's really in the Clavata pathway, there's a really neat assay, again, first developed in Arabidopsis, where you can actually synthesize Clavata 3 peptide. So remember, Clavata 3 is the ligand for these receptors. It's a 12 amino acid peptide. So you can just, ask GenScript to make it, and a week later you have the peptide. If you add that to plants, it will inhibit meristem proliferation, right? So mutants in these genes, the meristems overproliferate. If you add back the peptide in excess, you get the negative phenotype, which is the opposite phenotype, which is you suppress meristem growth. So basically, we do these experiments with embryos, because in maize embryos, they're big enough that we can dissect them and culture them, and the meristem is exposed, so we can easily access them with the peptide. So we culture the embryos on media with Clavata 3, or a control with a scrambled peptide. So it's the same sequence, but just the order of the amino acids is scrambled, so it shouldn't have any effect. And when we do that, we find that with the scrambled peptide, we don't see any difference between the mutant and the wild type. But when we treat these with Clavata 3, the wild type meristems, their growth is inhibited, as we predicted. The G-alpha mutants, CT2, they're also inhibited, but much less than the wild type. So we say they're partially insensitive to this peptide. And to us, this says that CT2 is somehow involved in transmitting this signal from the peptide to the meristem proliferation pathway. Okay, so to summarize this part, what we think is happening now is that, in animals and in yeast, this heterotrimeric G protein complex interacts with these 7 PAS receptors, these GPCRs. But in plants, it seems to be very different. There are GPCRs in plants, or at least proteins predicted to be GPCRs, but they haven't been convincingly shown to interact with G-alpha. But we found that G-alpha interacts with a Clavata receptor, Phe2. And so we published that a few years ago, and I was kind of nervous about publishing something that's so dramatically different between animals and plants. But since then, there have been several other papers showing that indeed G proteins do interact with LRR receptors in plants. So this is both in development and also in defense signaling. Okay, so now we wanted to look at, is this just an effect of G-alpha, or is this whole heterotrimeric complex involved? So as I said, it's a 3-protein complex. There's G-alpha, G-beta, and G-gamma. And we actually went after G-beta next for the obvious reason that it's a single copy gene. So mutants should have a phenotype. So, and the other reason was that work in Arabidopsis, published very soon after our work in Maize, actually showed that in Arabidopsis, G-beta controls meristem size. So we had G-alpha controlling meristem size in our mutants. In Arabidopsis, it's G-beta, right? So G-beta mutants in Arabidopsis have big meristems, and this group in Japan found that G-beta interacts with RPK2, which is not a classic Clavata receptor, but it's a Clavata-related receptor that also functions in the meristem. So it seems there's precedent that these G proteins, probably the whole complex, is functioning in meristem control. Okay, so this was our first CRISPR experiment. We did this with Ding Yang at Iowa State, and we were super excited. This was about 3 years ago. We thought we're going to get these plants back and they'll have big meristems. And we got them, and we were very disappointed that they were dead, basically. Right, so this, for developmental biologists, this is not a very satisfying phenotype. So basically, well, we could recover these because we got events back that were heterozygous, but when we selfed them, the homozygous Gbeta mutants, they germinated, and then within 3 days, they just dead, right? This is in stark contrast to Arabidopsis. Remember, I told you the Arabidopsis mutants have big meristems, but you know, the plants themselves look pretty normal, so we were really surprised. So we looked a little bit more closely at this, and what we realized is that the plants were not dying, were not just arresting, but they were actually becoming very necrotic. And we saw that cells were dying, the tissue was turning brown very quickly. And so we did some assays for hydrogen peroxide and for cell viability. And we quickly realized that, yeah, these mutants were actually undergoing some kind of autoimmune response where they were actually killing themselves. And we also looked at pathogenesis-related gene expression. So we think this is some kind of autoimmune response. And it kind of, well, it makes sense based on what I'll tell you next. But yeah, it's, we also grew the seedlings or seeds under sterile conditions, and they still died. So it's not a pathogen thing. It's they're just killing themselves. Okay, so this sort of makes sense based on what in Arabidopsis groups have looked at the G protein mutants in Arabidopsis, and Gbeta mutants actually are more susceptible to various pathogens. So this is mock inoculation of wild type Columbia, and then the AGB1, the Arabidopsis Gbeta mutants are much more susceptible to a range of different diseases. So it seems that, as I alluded to earlier, these G proteins are probably involved in signaling from defense receptors as well as from the Clavata development receptors. And in fact, a really nice story came out last year from Jiaming Zhou's group, actually showed that Gbeta interacts with FLS2. So FLS2 is an immune receptor in Arabidopsis that's involved in the innate immune signaling. And Gbeta interacts with FLS2, and that explains the phenotypes. And it actually seems to function as a positive regulator of immunity, but through its interaction with FLS2, but also by stabilizing a downstream kinase of FLS2 called BIC1. So Gbeta functions in immunity in Arabidopsis, and the mutants are more susceptible to diseases. But this really still doesn't explain why our Maize mutants are much more severe, right? Our Maize mutants are not more susceptible, they're just dead, even in the absence of pathogens. So we figure this out by thinking about how, how immunity signaling works in plants. So you probably are all much more familiar with this than I am, but as you know, plants are constantly bombarded by pathogens, and pathogens secrete effectors into the plant cell that try to suppress the cellular immune signaling pathways or defense pathways. And in the case of a viable infection where there's no resistance, then the plant is susceptible and will be affected by the pathogen. Now, plants have evolved these R proteins, resistance proteins, that can either interact directly with the bacterial effectors, the pathogen effector, but more frequently, it's thought that these R proteins interact with another host protein, another plant protein that is targeted by the effector. And they're actually guarding or surveying that host protein to see if it's under attack by a pathogen effector, right? So this is called the guard hypothesis. And these plant proteins that are bound by the R proteins are called guardees. They're being guarded so that when the plant is under attack by an effector, it knows it, right? And it's known that if you mutate these guardees, then the plant, again, thinks it's being attacked, and you get an autoimmune phenotype. So that's what we think is happening with Gbeta, that we think Gbeta is guarded by an R protein or an R gene. And when we mutate it, the plant think it's being attacked by pathogens, and it kills basically every cell. Now we can test that. One way to test that is that our genes are very polymorphic between different ecotypes. And so we took our Gbeta mutant as a head. Obviously, we can't cross the homozygotes, but we crossed the head to all of the NAM maize diversity lines and made F2s. And we found that for most of them, we still got the same phenotype, that the mutants were lethal. But we have a few lines where now the phenotype is suppressed, and we get plants back. They're not completely wild type, but they're not dead, obviously. They grow, they make inflorescences. So we think these suppressors are lines, ecotypes, that no longer contain the R gene. And in fact, we've mapped one of these, and it maps in a region. We don't know the exact gene yet, but it maps in a region where there is a predicted R gene. So this was a bit of an aside for us, but it could actually get us to a new R gene in maize, which would be cool, because actually very few R genes have been cloned in maize up to now. Okay, but anyway, we were excited to have these plants, because now we say this is now the developmental phenotype of Gbeta, and it actually looks very similar to G alpha, the CT2 phenotype. Now we've suppressed the defense responses, and they have, so we call these Gbeta suppressed, they have big meristems. And we think this is significant. And actually, coming back to the concept I brought up earlier, Gbeta in maize in GWAS analysis associates with kernel row number, the trait that I mentioned that we think is associated with meristem size. So we're still working on this, but we think, at least up to now, we think that Gbeta is also functioning in meristem signaling. Okay. So now we have a situation where Fe2, this Claddagh receptor, is probably signaling through G alpha and Gbeta. This probably G gamma also, but we haven't done that yet. Now, there's work in Arabidopsis that the ortholog of Fe2, which is Claddagh2, signals through a different protein. And that protein is called Coronae. Coronae is a kind, well, it was, when it was first cloned, it was published to be a kinase, but it's actually a pseudokinase. So it's a protein that has homology to kinases, but has no kinase activity, probably acting as some kind of scaffold. So, you know, I'm always interested in this question about how meristems evolve and whether the same pathways are present in maize, or let's say in monocots and in dicots, or whether they evolve or evolve separately or evolve to be different, right? So here we have one receptor, Fe2 in its ortholog in Arabidopsis, right? So presumably doing the same thing. They have the same phenotype, but that up to now, the genetics suggests that this one goes through G proteins and this one goes through Coronae. So we really wanted to know, is this a trans-species difference, or are both systems operating in both monocots and dicots? Right. So we were fortunate to get a mutant of Coronae in maize. We got this from Bob Mealy, a pioneer from their transposon collection. And we also got another allele from the amazing populations developed by Jerry Nufa, the EMS populations. And we found that Coronae indeed also is fasciated. So big meristems and a fasciation in the ears. So it seems that both Coronae and G alpha function in maize in meristem signaling. Now, again, we use double mutants. And so we have, I should have had a summary here, but we have Fe2, the receptor, and then two possible downstream signaling components, G alpha and Coronae. If we make double mutants between G alpha and Coronae, they're additive or synergistic. So we get these crazy fasciation that's much more severe than the single mutants. So this suggests they're different pathways. Okay. Now, however, whoops, there's a bit of a lag. Yeah. When we make double mutants between the receptor and Coronae, so between Fe2 and Coronae, here we see epistasis. So the double mutants look like the single mutants, right? So that's in measuring in meristems or just looking at them. They kind of look not that much different. So Fe2 and Coronae are in the same pathway. Coronae and CT2 are in different pathways. And our earlier published work had shown that Phe2 and CT2 are in different pathways. So this is going to be on the test. I know it's getting very complicated, but I'll have a summary. Okay, so what we think is the receptor has two different outputs, Coronae and Galpha. It turns out, and this is from some early work that we published that I'm not going to talk much about, that there are two different Gbeta-3 related proteins in maize that affect meristems. One of them is called Cle7, which we think is the Gbeta-3 ortholog. The other one is called FCP1. And we CRISPR'd both of these to get phenotypes. And we have fasciation in both cases. But what we find, and this is the important result, is that, again, we did these peptide sensitivity assays, where if a gene is involved in possession of a peptide, the mutant should be insensitive to the peptide, right? And we found that Galpha or CT2 is sensitive to one of the two peptides, but not the other. And Coronae is sensitive to the other peptide and not the first one. So yeah, so basically, this is our model. I know it's a lot of data, and it's even I get confused by this, but our model is that we have two different signals going through the same receptor, two different peptides. One is Cle7, which is the maize version of Gbeta-3, and one is FCP1, which is a different Gbeta-3 related peptide. Two different signals go through the same receptor, and they come out to different outputs. Now, you can ask me later how this works, and it's not that confusing because we think that these receptors do not work by themselves, but they work in complexes. So we think that Phe2 probably is a scaffold for other receptors which give the specificity. So maybe one receptor complex binds Gbeta-3, and another receptor complex binds FCP1. And we'd really like to know what those are, but we're not there yet. But at least phenomenologically, I think this is interesting because it shows how you can get different signal inputs and outputs going through a common receptor. So I think it's quite interesting. Okay. Yeah, I'm actually going to skip that because I'm the time, but it's probably running late. Yeah. Anyway, okay. So there's another area we're very interested in, which is looking at natural variation. And some of this is actually work inspired by work from Ed Buckler and Mike Lee and others, where they've shown that we can map QTLs for a kernel row number, which is the trait we eventually want to get to. So there is natural genetic variation in maize for, we think, for meristem size, certainly for seed number. And this is me in our summer field last year. This is actually a real corn plant. I didn't glue these together. And I'm the scale by here, so it's huge. This is a Mexican landrace from CIMMYT, the maize and wheat research center in Mexico. And I ordered these seeds, or seeds of this line, because they're annotated as being fasciated, and they are. And this is really amazing. This is a landrace that people grow and eat in Mexico, and it's amazing. And I asked my undergrads to shoot back these plants, and they were like climbing on each other's shoulders and bringing ladders. And then I told them you can just bend them over because they have stalks like about 6 inches thick, so they're really strong. But anyway, it was fun. Okay, so we are trying to map this, but we're not there yet. We're close. But another approach we've taken is to use the NAM population, which is this amazing resource in maize, natural variation resource generated by Major Goodman and made famous by Ed Buckler and many other people here. So this is a diversity population with 25 diverse founder lines. So we want to find natural variation in our trait of interest, which is fasciation. So we take one of our mutants, like Phe2, and we cross it to all of the diversity parents, and then make F2s and score the phenotypes. So we're looking for modifiers, genetic modifiers. And it's a lot of work, but it's kind of fun to just walk through the field and cut up in 10,000 years. It's one of my favorite activities, but not for everybody. But anyway, when we did this, we found that one of these lines, which is NC350, strongly enhanced the Phe2 phenotype, right? So NC350 by itself is not fasciated, right? These are just natural inbred varieties. But when we cross NC350 with Phe2 and make an F2, we go from a pretty strong fasciation phenotype that looks like this. So the meristem is really flattened and the ear is flattened. And then we get something like this, which is just completely crazy. It's really fasciated. It kind of looks like a brain. And I get my neurobiologist colleagues interested in this till they realize it's a plant. Yeah, so it's really strongly enhanced. So we want to know, you know, there's some locus in NC350 that's doing this. It's in wild type NC350, it's not doing anything to the meristem, but when we put it in a mutant background, which we think sensitizes the plant, then we see this really strong enhancement. Okay. So we map this in, actually this was originally really difficult to map because there's a lot of things segregating here, and it's probably not just one major locus, but we do several back crosses and then make an F2, and we found a major locus on chromosome 5 that maps to this trait. And we map this, and right in the middle is one of our favorite genes that I haven't really talked about yet, TD1, which is the Maize Clavada1 receptor. So Phe2 is Clavada2 and TD1 is Clavada1, so it makes perfect sense. A little bit disappointing, it's nothing new, but I think it's interesting that within this line, we've found that there's natural variation in TD1, which can enhance this trait. And we've confirmed this by mutants, and also, actually, I don't have it here in the slide, but there is a difference in expression in TD1 in NC350 compared to B73, which was our starting line. So it's interesting, and I think this is an extra resource we can hopefully use to uncover new, hopefully in the future, new loci also that control these traits. Okay, so last part of the talk, I want to just come back to this idea again about increasing yield, or at least increasing kernel number. So again, the idea is these mutants make massive disorganized meristems. The yield is very low, but if we can control this gene, i.e. a weak allele, we could make a little bit bigger meristems and more rows. Okay, so originally we did this together with a tilling facility in Maize set up by Cliff Weil at Purdue, and he screened for us in EMS populations for alleles of Phe2 that had point mutations generated by EMS, just by sequencing. We had 4 of these, and they're predicted to give single amino acid changes, so presumably weak alleles. And for one of these, at least, two of them, but one was more dramatic, we actually found that indeed, in these mutants, these weak alleles of Phe2, we do get an increase in kernel rows from an average of about 16 to an average of about 20, which is quite dramatic. A lot more kernels per ear, and everybody was excited about this. I wasn't really excited about this. I was really excited about this, which is when we look at these in the scanning EM, we see that indeed these weak alleles of Phe2, the meristems do indeed get a bit bigger, but they're still organized. Still this nice dome shape. They're not fasciated and splitting, right? So I was really happy about this. Okay, so we're making more kernels. Do we make more yield? Well, the answer is no, right? So as I was discussing with Bob this morning, you know, plants are pretty smart. You know, you try and trick it into making more rows of seeds, and they say, well, you know, I'm just gonna make the seeds smaller. So that's what the plant did. So there was no increase in yield in this background, which is not, you know, what farmers grow. This is W22, so it's not even a hybrid, which is what we normally grow. But, you know, we've been giving these lines out to breeders here and there, and actually a former postdoc in my lab is now a maize breeder in Vietnam, and he put this into their breeding lines that they actually grow, that farmers grow, and they again saw an increase in kernel row number. And in their yield trials, this is real breeders' yield trials, this is not our crappy field in Long Island, they saw up to a 20% increase in yield. So that's pretty encouraging. It needs to be reproduced, but I think in some backgrounds, this may really have an effect. We did this with another gene that we worked on called Phe3. Haven't really talked about this today. In this example, we had weak alleles. See, they're still mildly fasciated, but nothing like the null mutant. Again, these are EMS weak alleles. In this case, even in our, you know, old inbreds and hybrids, it's just B73W22, we see a pretty significant increase in yield. Again, this is not elite varieties, of course, but encouraging that we may do this. So maybe these meristem pathways are interacting somehow in pathways that control zinc strength, right? So the plant only has a limited amount of resources, but what it does with that resources, I think, is a mystery, how that's decided. So there may be some interaction. Okay, so we want to do this large scale. And I think with the EMS alleles, we were really lucky to get alleles that had the desired effect. So we want to make a more generalizable approach to make weak alleles in plants, right? And targeting the protein sequence is a little risky because you might, you could knock out the whole protein. You don't really know what you're going to get, and EMS is kind of messy and slow. And so now, of course, we have CRISPR, which is this amazing new tool. So to make weak alleles, we don't want to target the coding sequence, but we thought maybe we can target the promoters. So again, this is with our CLE mutants in Maize. What we do is we take the predicted promoter region, and we don't care about promoter elements. Everybody asks, like, how did you predict which promoter element is important, blah, blah, blah. I don't care. I just want to get a phenotype, right? So we take the upstream 1 to 2 kb of promoter and just tile it with 8 or 9 guide RNAs. Then we transform this into Maize, and what we get out is plants that have all sorts of different genotypes. So basically, we have small indels, which is very common with CRISPR, but if you do CRISPR with multiple guide RNAs, you also often get large deletions. So we get different promoter deletions. So this is like in vivo promoter bashing. I mean, when I was a graduate student, everybody was doing promoter bashing, and I thought it was really boring, but I'm actually quite excited about this because it's in vivo in the native genomic context. And I wish I had a lot of results from this. You may have seen a paper published a few months ago from Zach Lippman. So we're doing this together with the Lippman lab. He's doing tomato, and tomato is much faster than Maize. And they already have amazing allelic variation in tomato. The only thing I can show in Maize, which is not very satisfying, is that we do have some weak phenotypes, some weak fasciation, but no yield data yet, but that's coming next summer. So I think it can work. I mean, there's no reason it shouldn't work. So I think it's pretty exciting. So where are we? So I think I've hopefully convinced you or confused you that the Claddagh network is very confusing and complex, and CRISPR is just beginning, so it's going to get a lot worse. We used to be in this great situation where we had, Claddagh 3 binds Claddagh 1, and that regulates Bush1, there's feedback, but we now know that there are many more receptors that function in meristem control, and then all these downstream signaling components. And there are a lot more that the paper I just reviewed has 4 more over here that will be coming out soon. So yeah, there's a lot more stuff coming. So it is very confusing, but in a way that's biology, and it's satisfying. I mean, it's interesting to think about why it's so complicated. We could discuss that later, but yeah, it is complicated, but we're making headway. And then there's the CLEs. So Claddagh 3 is this, as I said, is this CLE, it's this peptide. It's a member of a big gene family. There are actually 49 in Maize. And I didn't dare ask my postdoc to knock out all of them, but he's actually done about 20 of them already. So this is amazing, not all in the same plant, right? So they all have to be crossed together, but the amazing power of CRISPR, we're now knocking out 4 genes at once, and we could go to more, I'm sure. So it's really, things are going to change very quickly. So we're focusing on only about 30. So I decided to be more modest in my expectations, but the ones that are most highly expressed in fluorescence, of course. And I'm sure there'll be lots of interesting phenotypes coming out of this, probably more than we can ever study. But I think it's very powerful now to think about, not looking at single genes, because we know that redundancy is very prevalent in plants, and so we need to think about knocking out whole gene families to find phenotypes. Okay, and then, so how complicated is it? Very. And then something I think about a lot also is, to what extent is crop yield limited by zinc? And when I talk about this, a lot of people say, well, yield is source limited, right? The plant only makes a certain amount of photosynthate, and all that goes to the ear, and then you can't make more by making the ear bigger because you only have a certain amount of resources. But I don't know if that's true. And in some of our work suggests it may not be true, right? I mean, the resources, maybe some of the resources are kept in the roots or kept in the leaves, and we're funneling more of that into the ear. And whether that's good or not for sustainability, I don't know, but it's interesting to think about whether this can be used to enhance yield. Maybe this has already been optimized in corn belt maize, because breeders have been breeding maize for almost 100 years, but probably not in all environments, as I showed you with the lines from Vietnam. And certainly there's a lot of interest now in developing new crops, right? Often crops, improving them, and I think these pathways could be very useful in improving yield in crops that are not common row crops now. Okay, so yeah, I just want to acknowledge people who did this. These are, in green, are the people involved in Claddagh-related projects. The other people are doing different things. A lot of the work is done by a very talented postdoc, Byung-il, who just got a job back in Korea, and also Peter Baumert, who's back in Germany. And then we have a great group of collaborators and funding agencies and et cetera. Okay, and thanks very much.
Question? I have two questions.
Okay.
So what you call yield is actually the weight of the ear.
Yeah.
Does it translate to, how does it translate to yield per acre?
Well, that depends on how these things do at high density, which we have not measured yet, right? So that's, but it's ongoing, but yeah.
And my second question is, have you ever looked at signatures of selection around any of those genes?
Yeah, I mean, I didn't go into this, but a lot of these genes come up in QTL scans or in GWAS studies. So yeah, so there's variation that's associated with yield traits.
So
or whether they're selected. Yeah. So yeah, it's an interesting question. So these genes did not come out in the Dobley screen, right? So Dobley crossed ancient maize and teosinte and found major QTLs, 5 major QTLs. So these genes are not in those. There is one interesting gene in there, actually leafy, the maize leafy homologue, which he showed maybe also involved in controlling row number. Yeah, so I think of these genes as not in the original domestication events, but more in improvement events, the more recent evolution. Yeah. So they do not come up in the Dobley data set. TD1 actually came out of the work, the paper published last year, I think in Current Biology, that Jeff Rossibara was on, where they sequenced ancient maize from these 5,000-year-old cobs, and they found that TD1 was already under selection nodes. So, yeah, I think it's there, but we haven't looked too deeply into that. Eric?
So you're using kernel row number as a proxy for meristem size or fasciation. Have you seen any examples where you see essentially more kernel rows, but the meristem looks normal?
So we actually, it happens, and obviously kernel size can vary, so you can get more rows by having smaller kernels. Originally we looked at all the NAM founder lines and tried to correlate kernel row number with meristem size, and I think meristem size variation explained about 60% of the difference in kernel row number. So it's not all of it, but it's a substantial part of it.
About your model of the two receptor complexes.
Right.
Gbeta and CRN. So the association of Galpha with the receptor is that direct. So then the prediction would be that probably it's through another.
Right. So yeah. So the question is about this sort of branching receptor pathway and whether the receptor interacts directly with Galpha. So we published that paper in 2013 that the receptor interacts with Galpha, and then Peter went off and got his job, and he left the lab, and then another postdoc joined and could not repeat it. So I was really nervous for a while, but eventually got the conditions right, and he repeated it. So it is true, but we detect the interaction by co-IP, but not by FRET or BIF-C. So we think there's another protein that bridges that interaction, and we think that is a receptor, but we haven't been able to purify it yet. We have a candidate.
So it seems like you've got lots of parallels between Arabidopsis and Maize.
Yeah.
Are there some real differences in the signaling pathways?
So I think Maize is somehow more sensitized. So I think we found a lot of things in Arabidopsis, in Maize, that have not been found in Arabidopsis, like Fe4, Galpha, and Fe3. Then when we've gone back and looked more carefully in Arabidopsis, we've found the phenotypes, either by making double mutant or, so we haven't seen anything that's really obvious. And I'm kind of happy about this. I have this sort of friendly back and forth with Elliot Meyerowitz that he always, he's not listening, right? He has a bit of an ego, let's say. So he'll just, if we find something in Maize that hasn't been found in Arabidopsis, he'll just say, oh, well, you just show that things are weird in Maize. But I don't think it is. I think they're the same. It's these basic pathways.
Let's thank David again.
This has been a production of Cornell University, on the web at cornell.edu.